Our next speaker is Professor David Haussler of University of California at Santa Cruz. As Mike mentioned, David was one of the leading participants in the assembly of the first draft of the Human Genome, and he leads the development of the UCSC Genome Browser, which everyone in the community uses. It's really inconceivable to think of doing bioinformatics these days without this. It's, I don't know if I'd call it the Wikipedia of bioinformatics, but definitely it's out there. It's maybe the Chrome of bioinformatics, Chrome browser of bioinformatics. So it's really an essential tool, and it's now available not only for a multitude of species and a huge amount of data types, but equally important for cancer projects. And the compilation of cancer data is crucial for breakthroughs in future cancer development. In addition to these, Professor Haussler was one of the first to introduce machine learning into bioinformatics, and by now they are ubiquitous in the field. He has won many awards. Let me just mention that he is a member of the National Academy of Science and a recipient of the Howard Hughes Investigator Award and of the Weldon Prize. Please welcome Professor Haussler.
Thanks so much, Ron. Is this working? The mic? Oh, I'll hit the button. Okay. Yeah. Well, it's really a pleasure to be here, and I want to thank everybody from the Daviv Prize, but also it's a huge pleasure to be here at Tel Aviv and get to experience this powerhouse of bioinformatics. And I thank Ron for the invitation to have a chat with you here about some of the latest things. So we're working at utilizing the amazing information that's now available from the advents in human genome sequencing that Mike was talking about. I'll give a little reminisce at the beginning. So in 1990, the Human Genome Project was launched, an international effort to sequence the heritage of the first of a single human being for the first time. And the project was dramatic in scope, and that it was building its own technology as it went along. It was planned to finish in 2005. It was very exciting in terms of its international scope. This is a picture of the Beijing Genome Center. At this time, Huaming Yang was leading that. In fact, this goes on quite a bit. I can't fit it all on one slide. It was a remarkable worldwide effort. But as you know, and as Mike mentioned, in 1998, 8 years into the project, Celera announced a faster, better way to sequence genomes, and said that they would finish in 2000, 5 years before the public project. And a lot of this has to do with the brilliance of this person, Gene Myers, who invented the whole genome shotgun method. and some ample funding to build a really huge sequencing center and a huge computing facility. Actually, they had one of the facilities that was close to one of the most powerful because Gene's efforts to assemble the whole genome shotgun reads, as Michael so brilliantly outlined, was an enormous computational effort. The public project, meanwhile, had planned to finish in 2005 and had not really had any infrastructure for the computational assembly of the Human Genome Project in the plan. So when we jumped in, we were actually supposed to do the human gene finding, because I had developed hidden Markov models to find genes, and we were going to assume that the thing had been assembled and we were going to find the genes. Well, that assumption didn't come true, and so we had to work on assembling it as well. So I actually lobbied the dean and the chancellor for some computing power, and this is our computer cluster. It's 100 Dell workstations, Pentium 3 processors, none as powerful as your cell phone is today. And we struggled to try to put together these pieces. And it's a crazy story because one wouldn't have imagined that it would happen this way at all. We actually, on May 7th, we heard in our weekly calls from Francis Collins that a deal had been struck with Craig Venter, and we would both be jointly announcing the first draft of the Human Genome on June 26th. We were on the call thinking, what? Are you serious? We are nowhere near actually assembling and providing for the world an entire human genome. So the real hero here is Jim Kent, a graduate student, nonetheless, at Santa Cruz, who wrote approximately 20,000 lines of C code in less than 4 weeks, and it put together about 600,000 DNA pieces that were left behind from this rush of the public project to get the sequencing done as fast as possible and meet this White House deadline. He literally had to ice his wrists at night. I would go over, he worked night and day. It was really, he'd take a nap and then work and then take a nap and then work. It was insane. He's very, very brilliant when it comes to software. And I often think back, if you'd had a team effort at this point, no way, right? With that amount of time, it had to be one person. There were actually 13 different inputs to this code, too, not just the DNA, but the genetic maps, the radiation hybrid maps, the physical maps, the gene sequences themselves, the RNA that spanned multiple. So all different pieces of information that were saying it should go one way or it should go another way, and he had to adjudicate that. So this is the New Yorker cartoon. He's looking at this 3 billion, close enough to 600,000 piece jigsaw puzzle. Says, I think I found a corner piece. So on June 26th, the grand announcement was made. Humanity had sequenced for the first time our genetic heritage, a message in DNA that had been passed from parent to offspring throughout the history of life on this planet, and now was our heritage. The most exciting for me, though, was a few weeks later on July 7th, when we got the opportunity, because we had assembled it, to actually post that on the internet. And I think back to that day, I actually sat and just to see whether the process was going on, watched the A's, T's, and G's go flowing across my screen, and I thought, now the human genome has been replicated in a way that nature had never intended. So it's being broadcast at light speed to all corners of the globe. And that, in a sense, is a portal for science and technology. We stepped through a portal there now. We can read our own genome for the first time. And we had that as a product of two great technologies of the latter half of the 20th century, the biotechnologies and the information technologies, and they really work together for this. This little chart on the bottom is the green is total outgoing internet traffic from Santa Cruz campus. So you can barely see it on the scale, and then we posted the genome and broke all records, about 0.5 terabyte in the first 24 hours. So while all this was happening, the world was getting its first glimpse of our genes, our mechanisms for development, starting to think about how we get somehow from a single fertilized egg to an entire adult based on what's in this sequence. The rest of us have been trying to figure out how to interpret it ever since then, and we're making some progress. So one tool, as was mentioned, is Jim actually went on to lead a team. He now has 30 people working in his team to build the UCSC Human Genome Browser. And it is used, this is a nice map of usage levels, but it's up to 1.2 million page hits a day at this point. It's really had a dramatic impact on the field by giving people, this is all about information, the Wikipedia, the theme of this, it's all about giving people easy access to integrated information from all of the different sources so that they can do their own little experiments. Oh, let's see if I look at this track of information juxtaposed against this track, I see a pattern, you're off and running with a hypothesis. It looks like this. I won't go to any details, but you can select now among many, many hundreds of different types of information, and they're all aligned along a one-dimensional map system, along the chromosome. So it's like Google Maps, but on a one-dimensional world. And now we just released genome hubs, so you can make your own hub with your own tracks of information very, very easily, and we've had more than 1,000 hubs that have been born at this point around the world, so we're very excited about that. The human genome is one of many genomes, and so one of the things that's incredibly important is that we have to understand our place in life as just one of the animals on this planet, and our genome history is intertwined with that of the other species on the planet. And we won't really be able to decode our own genome without understanding those other species. So several years ago, I worked with Ollie Ryder and Steve O'Brien to encourage the scientific community to maintain funding for comparative genomics. And in the US, that's been a hard slog, actually. You may have noticed that the NIH funding for comparative genomics is kind of dropped off as it began to fund other things. So I still think it's very important that we make the global effort. Part of this is to sequence species that are endangered for purposes of species conservation and making sure that we are good stewards of this planet. Our goal of sequencing genomes from 10,000 different vertebrate species is just one kind of stake in the ground. There are also other projects that are very similar, sequencing large numbers of species from other clades. And it's important that we go on and do this for understanding and for conservation. We actually heard at the Genome 10K meeting in Santa Cruz in March that the conservation uses of this are finally actually happening. They have controlled breeding now based on genome sequences in cases that you only have 7 or 12 or 13 members living of a species. You want to have the optimum breeding strategy, so you sequence their genomes. One of my favorite other parts of the newest applications of genome sequencing technology is not only sequencing living species, but sequencing extinct species. And my colleague Ed Green was first author on a very high-profile paper with the first sequence with Svante Pabo of the Neanderthal genome. And In that paper, the kind of surprising announcement is that non-Africans actually have a reasonable portion of their genome that they got from Neanderthals. So the combination of sequencing living and extinct species gives us a remarkable feel for the diversity on the planet, that even within humans, there is an amazing genetic diversity. So we now, with a stunning drop in the cost, as illustrated on Michael's slide, have an opportunity to sequence an immense diversity of human genomes and understand our genetic heritage. So just like when we look at an endangered species, we don't want to just sequence one individual. We want to know the total amount of genome variation that is left in that species so that we can preserve it all. Actually, we need to take the same approach to our own human species and understand the total amount of genetic divergence that is exhibited, at least any variant that's segregating, say, at 1% in a well-defined population is something that is clearly an important part of our genetic heritage. Now, the problem that we have encountered with trying to understand and make easily accessible and doing analysis on our human variation is that there are a hell of a lot of different ways of looking at it and representing it. So these are just some of the databases on which you can find variation. The 1,000 Genomes Project has probably the premier site, but we have UK 10K and now 100K. dbSNP is a very, very popular resource, but it only includes some types of variation, and it has its own format. There's a database for genomics variants. There's ClinVar. So one of these issues that comes up immediately in this is not only in the number of different databases, but the number of different nomenclatures and symbolisms for this variation. So it becomes a huge Tower of Babel. Who's actually talking about what variant? Is it the same variant? What are the total number of variants? So we have set out recently with an organization called the Global Alliance for Genomics and Health that I will talk more about later to try to make some kind of Rosetta Stone for all of these different ways of representing human variation. And we're starting from the assumption now that we can take advantage of whole genome sequencing in a way that was never possible earlier. When we think about if we had thousands and thousands of whole genomes, how would we represent that data? Well, you can think about it as squashing them into a graph. And this is a picture I took from Gil McVean. This, ladies and gentlemen, is a view of your major histocompatibility complex, the MHC region of chromosome 6, which is fundamental in your adaptive immune system. Every one of those lines represents a different person's MHC, and they converge or superimposed on each other when the DNA is similar, and they diverge when the DNA is divergent. So this is just one cut at how we might think about the variation that that exists and is segregating in the human population in one little region of the human genome. Our current reference genome may be just the red line. So that is not really capturing all of the diversity that we need. How do they fix that? Well, they added to the reference 7 other variants of this. And unfortunately, if you are a bioinformatician, you know that almost everybody ignores those at this point when they map their reads and so forth. They're so addicted to the one true human genome with one version of chromosome 6 that they just ignore the rest of the version. This is crazy. This will not, you get bias in all of the analysis from this. So what do we do if you take an object and we can actually compress thousands and thousands of genomes, a representative set of human genomes, into a graph like this? What's underneath the hood? So if you zoom in, you would see segments of DNA in more precise levels. And during this little part of the graph, there's basically a human population that does something like the yellow things, and another population that's doing kind of the red and the green thing. And within those, there are minor little variants, right? So there's all kinds of micro variation and macro variation at different scales. If we zoom in a little further, we can actually get down to the base level. We propose a hashing scheme system in which every one of these individual bases would have some kind of universal identifier. So this graph that we would share, we would build and share, would be a reference that in which the coordinates weren't numbered from 1 to 2 along the one chromosome, but you would have an identifier that you could get an access to for every position. Probably not a UUID, that's too many bits, but we'll have a hierarchical system for that. Now, the nice part about this is, suppose at some point later we decide that actually we want to change the graph, say by adding another G up here, then the identifier for everything downstream or in any other place, and that doesn't change, right? Which is one of the fundamental mistakes we made with the way we currently do the genome, right? Every time we get a new assembly of the human genome, all the coordinates change. So this approach is fascinating, and we're in the throes of trying to figure out how to work it out. I'm going to move on now to another subject which is very near and dear to my heart, and I'll finish the talk looking at cancer. And here my colleague Josh Stewart is playing a major role. Josh is actually leading the pan-cancer comparison, first for the US Cancer Genome Atlas, and now for the International Cancer Genome Consortium as one of 5 international leaders. This is our first opportunity to really take a lot of cancer genomes and compare them in detail with all of the best bioinformatics methods. In order to support the Cancer Genome Atlas of the National Cancer Institute at the United States, our team actually built another resource, and this was kind of deja vu from the first, from the first assembly. They had some plans for storing all of these thousands and thousands of genomes, and then it really wasn't working out, so people couldn't get access to the data, and it became a nightmare. And so we stepped in and we built an entity called Cancer Genome Hub, or CG Hub. That actually is now the most active transfer of cancer genome information in the world. We're actually shipping I think it's 1.3 petabytes of information out over the web to researchers all around the world every month, so over a petabyte a month. That's more than NCBI more than all the downloads from NCBI, actually. In order to do that, we became the first trusted partner of the National Institutes of Health, which is a funny status, but it means that you can redistribute data, even restricted access data, such as personal cancer data. And so this was part of opening up of these data so that the bioinformatics community has easier access to them. You still have to register and apply for access and prove that you have the credentials. When we looked in a team led by Josh Stewart at all of the different cancers, they're classified according to their tumor of origin. So just symbolically here, we have bladder cancers, lung cancers, liver cancers, brain cancers of various types. And if you look at the molecular features and map the tumor according to its molecular features, you see a dominant clustering based on the organ of origin. So it is true that most of the molecular information in a tumor talks about what organ it came from. So you have this nice clustering of lung cancers here and this nice clustering of bladder cancers here. But then what was very exciting were the exceptions. Oh, here's an individual with lung cancer, and molecularly that lung cancer looks a hell of a lot more like bladder cancer. So this is exciting, and in fact, they found in the first one with TCJ that about 5% of the samples really had a different molecular classification. And that's important because we're thinking about molecularly treating these. This is actual data out of this. It's a two-dimensional projection kind of with a Google Maps hexagon technology. So every little dot in the hexagon is an actual data point in the Cancer Genome Atlas. And every odd-colored piece is something that's molecularly different from its tumor of origin. One of the most important things to think about today is childhood cancer. And the National Cancer Institute has a separate program for this. So the Cancer Genome Atlas is about 30 adult cancers, and there's a program called Target for childhood cancer. So we added all the childhood cancer genomes to our Cancer Genome Hub, and we became very, very determined to do something about childhood cancer. Actually, when this is the daughter, Aurora, of one of our researchers in our lab who got neuroblastoma at this age. She's fine now. She recovered. But it was really a wake-up call. The statistics are really frightening. So 1 in 300 people will get cancer before age 21. I was shocked to find that, and it seems like we should all drop everything really and try to make sure that that statistic is improved. We've come up to actually 80% in terms of a success rate, but if you look carefully into that, the success often comes with lifelong disabilities because childhood cancer is treated incredibly aggressively. They throw the most powerful chemo, radiation, everything in the book to try to save the child, and those can unfortunately, in almost half the cases, have lifelong sequelae. One of the most important things to think about in childhood cancer is this is a rare cancer. Most childhood cancers are extremely rare types of cancers, and so we don't have enough information in any one institution to really understand. Often it may be the only case that institution has seen. So the most important thing is to share the data between the institutions, and we've managed through our TREEHOUSE childhood cancer collaboration to get 21 different institutions to agree now to share the genome sequencing data that is increasingly available in childhood cancer. And we're using our network and computer capabilities and so forth to provide analysis and support. I'll go into this now in a little bit of detail. Where is my timing? So one of the most devastating childhood cancers is glioma, and the childhood versions of it tend to be in this side called lower grade glioma, and then the adult versions are glioblastoma, and unfortunately the lower grade gliomas often later in life progress to glioblastomas. These are extremely difficult tumors to treat. I'll go zoom in on the glioblastomas a little bit to illustrate some of the data and approach that we take to those. First of all, we start with sequencing machines that read short pieces of DNA cheaply. We would love to have longer reads to sort out the rearrangements and so forth, but right now we're dealing with short reads. We can map them onto the reference genome. We would like, again, to map them onto a richer reference structure, but we map them onto the reference genome. And we note that places where unusual things happen. First, you can look at the depth of reads, and there's a block of 0.5 megabase in which we have extra read depth that indicates that the tumor has an extra copy of that stretch of DNA. And you can actually zoom in on these reads, and you can find a place where what's normally chromosome 2 at about 29 million goes along and switches from red to blue to chromosome 2, 28,500,000. So it's like it's jumped back on itself. We were reading here in the genome, and then we jumped back and we started reading here. This suggests a tandem duplication of this region of about half a megabase, and that's exactly what's going on. And what's amazing about this is that we've nailed a half a megabase tandem duplication right down to the base, exactly where the breakpoint is. You can put together all of these breakpoints, and in this Circos diagram, we're just looking at reference chromosome 16, and we're looking at cases where there is a breakpoint with these red arcs, meaning that there's a place where part of the DNA reads is here and the other part is over here relative to the reference. And if you look into this in detail, you can actually take this and walk through a version of the tumor. So here's the standard reference human genome. There are 5 regions, 1, 2, 3, 4, 5. We have tumor reads starting here that jump over to this side of 4 and then jump back 2, 3, and so forth. And so if you walk these connections, you see that the prediction is that the tumor will have the first segment, then an inverted version of the 4th segment, then the 2nd segment, then the 5th segment. And it skips over 3 according to this path analysis. So immediately you check the copy number and you see this. Well, that confirms that there is a missing copy of 3. So we have an explanation of what's going on, and we can actually go to the Human Genome Browser and look for what genes are. This is the normal configuration of that version of the chromosome, and in this cancer, it looks like this. The main part is flipped around, and some parts are missing, and you can see what genes are disrupted. You can even see whether other genes now maybe have different upstream regulatory signals. If you look at glioblastomas, you see an amazing amount of this kind of change. And so here's a glioblastoma in which when you walk through the different connections like I described, you start at this part of the genome, you go down here, and then you go down here, and so forth, and you come all the way around to back to where you started after going through about a megabase of DNA. So we get a circular map of this portion of the tumor genome. And the remarkable thing is that this is real, and it's called a double minute. So we actually have within the glioma cells a small circular chromosome that's in this case highly amplified, and often is highly amplified. And it's the pink background. If you do actually some fluorescent in situ hybridization, you can see that there are many, many copies, whereas of a normal thing, there's 2 copies per cell. There's many, many copies of this per cell. And it contains important markers, like important genes like MDM2, which is an inhibitor of P53. So when you amplify MDM2, you inhibit P53, and that loses the protection that P53 provides from cancer. Here's another example from a different one. In this case, we found it in a lower grade glioma. So what the next few slides show is that things we saw from the high-grade gliomas, the glioblastomas, the deadly adult cancers, we started to see some similar things in a portion of the lower grade gliomas, suggesting that there was a molecularly distinct subclass of lower grade gliomas that act like glioblastomas. So here's a double minute we traced out of a lower grade glioma. They weren't thought to occur very often in there. When we were looking at these and other lower grade gliomas, we found that another hallmark of glioblastoma showed up, again, somewhat unexpectedly. So whereas P53 is kind of like the brakes that keep you protected from cancer, and so when you lose P53, it's like not having any brakes. These genes, receptor tyrosine kinases, are accelerator pedals, and they stimulate the cell cycle and the growth of the tumor. And so if you have a version of these where they're amplified or even stuck in the on position, then you have a stuck accelerator pedal, essentially. There are many ways that this can happen, and it happens in almost every adult glioblastoma. But again, it was surprising to see it happening so often in these lower grade gliomas. And what was notable is that we saw in the lower grade gliomas that didn't have this mutation of the gene IDH1. Now, Bert Vogelstein was the first to discover that this was a very important gene, and almost all of the lower grade gliomas he looked at had this gene mutated or its paralog. So lower grade gliomas that don't have this gene mutated are called IDH wild type or IDHWT. They're actually rarer, and it was these that looked like glioblastomas because either EGFR, one of the most important RTKs, or one of these other receptor tyrosine kinases was mutated. You can see the way that they're mutated happens in different sources. It can be from amplification, indels, single nucleotide variants. You see that it happens more in higher grade. And actually, many examples have multiple mutation events. So there are different routes to amplifying one of these receptor tyrosine kinases. One of the most fascinating ways in which it occurs is gene fusion. So this is known in cancer that you create an abnormal gene by fusing pieces of two normal genes. And this is conceptually what this process looks like. We discover this by RNA sequencing often. So you can find an RNA transcript that clearly is a funny chimeric transcript. With short reads, you're looking for kind of spanning reads. And if you're lucky, you can also validate this in the DNA. So what we found, I'll go through these really quickly, is a kind of a gallery of strange variants of EGFR. This is the business part of EGFR on this side. And then we found out, and this is the control region for it, we found that was chopped off and replaced by something else. And it didn't seem to matter a whole lot about what that something else is. In fact, there are some patterns in what gets replaced here on this side for functionality, but the main thing is that the normal control region is gone. And so you have a stuck accelerator pedal here. There's no way that the cell can control EGFR anymore. This also happens with another tyrosine receptor kinase, FGFR3. And again, there's something of a pattern to its to the other gene that replaces its control region, but it's subtle. So you start to see that there is a subclass of lower grade gliomas that have molecular characteristics of high grade glioblastoma. What does that mean for actually treating gliomas, to treat brain cancers? Well, if a young adult, this is very common in ages 20 to 35, comes up with a lower grade glioma, currently the histologist will look at it under the microscope, and based on what they see, they will determine whether it is any of these three types: astrocytoma, oligodendroglioma, or oligoastrocytoma, which is just kind of a mixture of these two. Based on that, you will get certain treatment. Now, after looking at many hundreds of these genomes from molecular detail, the lower grade glioma working group of the Cancer Genome Atlas, which we participated in, was able to come up with a much better classification. In particular, they confirmed Vogelstein observation that, in fact, the IDH1 was extremely important. Out of 282, there were only 58 where it wasn't mutated. So these wild types are a special category, and there's where we saw the EGFR and some other characteristic mutations from high-grade gliomas, glioblastomas. And of those that did have the mutation, there were some that also had a second mutation, a loss of the P arm of chromosome 1 and the Q arm of chromosome 19, and then those that didn't. And these had very distinctive, different molecular signatures. And so out of this analysis, and we actually did this independent clustering based on 3 or 4 different data sets, and it always came up with these 3 molecular classes. So these are quite robust molecular classes. Now, it makes sense that these might be better prognostic classes. So we also have data that says how long the patient survived in terms of years. And if you use the normal histology, so this is histology type, astro, oligo, and so forth, and this is histology grade, grade 2, grade 3. The difference in survival is not so apparent. So as you go across this Kaplan-Meier plot, the percentage that has survived that amount of time drops here down to 20%. If you use the molecular classification, you see that there's this one class, this red IDH wild type that looks molecularly like adult high-grade glioma, glioblastoma, and you see this horrific drop to or about 25% survival in about a year, as compared dramatically different to the behavior of the other 2 molecular types that do have the IDH mutation. So this distinction is much more important than what you see in the microscope. This is another validation of that. Now we've actually looked at actual adult GBMs. So we have adult GBMs, which out of approximately 400 adult GBMs, 373 were wild type for IDH. That's characteristic of the adult GBMs, and you see their steep drop-off in survival. And then there are these rare 24 cases of these odd adult GBMs that didn't have the mutations. Some of those we don't know whether they're mistakes or not. They have a funny little plateau here in terms of survival that may be real. But what's interesting is the low-grade gliomas that we found that were wild type cluster right in here with the high-grade gliomas. So the conclusion of this is that lower grade glioma with IDH wild type is glioblastoma, period. You do not have the more benign version, and you need the most aggressive therapy for that. And this is now becoming official. So the World Health Organization, based on these data, is changing the criteria for treatment, standard treatment of glioma, which is the ultimate satisfaction in doing this kind of scientific work. I want to say a few things just in ending here. One of the most exciting things that is now occurring in cancer treatment is immunotherapy. And the idea is to train the patient's own immune system to attack the cancer. Many, many cancers we're finding are already recognized by the T cells of your adaptive immune system, and they can distinguish the cancer cells from the normal cells. But either there aren't enough of them, or they've become anergic by some clever signaling that happens from the tumor. So the tumor is hiding from the immune system in some way. We know that this is going on. We're working with Steve Rosenberg, who actually takes T cells out of the patient, creates and expands them into up to 1 billion different T cells that are targeted against the tumor, activates them by giving them cytokine signals, and injects the beefed up T cells back into the patient and gets really amazing results. So that's a direct proof that if you enhance the immune response to cancer, it will take care of the cancer. This is very dramatic, especially in melanoma, where we almost always have a T cell recognition. And one of the reasons for this is that melanoma carries an enormous number of mutations. And so those mutated peptides are displayed by the HLA molecules in the MHC region, and that gives away the fact that the cell is a cancer cell. Now there are a couple of drugs that will do this much more efficiently. They'll block the signaling. They won't allow the cancer to shut down the T cell response, and they get spectacular results. Those were definitely the most exciting results at the American Association for Cancer Research in Philadelphia in April. 6,000 scientists, they could barely get them in the room waiting to hear the results from the latest immunotherapy trials. It's very, very exciting and a lot of potential. And this illustrates, the last point is that one of the main problems in treating cancer, even with precision medicine, is that cancer, in a sense, undergoes its own little evolutionary process. So here is a patient with metastatic melanoma. He's got a mutation in a gene called BRAF, which is kind of like EGFR. It's one of these accelerator pedals. And this mutation makes it kind of like a stuck accelerator. There's a drug, vermorafenib, that will actually kill any cell with a mutant form of BRAF. Patient takes the drug, and 15 weeks later, amazing. I mean, it's really unbelievable. This definitely made headlines when it happened. But unfortunately, in every one of these tumors, there are many, many cancer cells, and we have a clonal evolution process that's going on. So as cells were accumulating more and more mutations and getting more and more aggressive in the cancer, different clones, showed by this shade, were expanding. So not every cell in every tumor has the same genome. So there were cells in most of these tumors that didn't have the BRAF mutation. It may have been 1/10,000% of the cells, but that's all it takes. So with a one agent that only kills that one vulnerability, the cells that didn't have that mutation grew back out. And what shocks me is this: if you look at this, it's like a map. There's kind of 3 tumors going across here. They all grew out. Some of the tumors didn't grow out, but most of them had some kind of resistance, and they eventually grew back. So we've seen this before. So if you think about our approach to HIV/AIDS, right, when we first gave one drug, the virus adapted. You gave it 2, it still adapts. You give it 3, it's too many, you're blocking too many things at once, and it's very unlikely that it will be able to simultaneously escape all 3 modalities of therapy. It would be wonderful to do that in cancer, but you give me 3 strong cancer drugs at once and it's going to kill me, unfortunately. So there is an issue here. One of the great things about immunotherapy is that the T cells actually recognize multiple epitopes on the cancer cells. And in fact, they themselves evolve, right? Because they have billions of years of training to try to fight off viruses and bacteria. They are remarkably diverse in how they will attack a tumor once you get them activated. So these things become a new modality. You could try a drug and an immunotherapy. You can try, if you want the 3 drugs, you can try cycling them, one drug for a while, then one drug, then one drug, and then back to the first drug if they're lethal if you take them both at once. So these new ideas are really exciting, and I want to end on a positive note. People say, once they learn about this adaptive thing, I've heard evolutionary biologists say, oh, cancer's hopeless because the cancer itself evolves as you treat it, right? Well, there's a big difference between an infectious disease and cancer. An infectious disease like a bacterium or a virus is its own organism. If HIV becomes resistant to our current therapies, we will have to find new therapies. And it will, right? Because it passes on its genome to its offspring. And there's an unbroken chain through time of this bacterium or this virus. And it will always be there, and we'll always have to come up with a new idea. There's no unbroken chain in cancer. Whatever happens in the body of an individual in kind of adapting to our therapy, all that knowledge that the cancer gets about how clever I beat this therapy dies with that individual. And then if you look at their children, it's the same game again. It never gets smarter. So once we realize how to win this battle once, we'll never have to think about it again. we will eventually defeat cancer much more soundly and completely than we will defeat viruses or bacteria. It's not really an evolutionary process. So in order to do that, we formed the Global Alliance for Genomics and Health. The key thing is sharing the data. And I won't go into a huge amount of detail here because I'm over time. But we now have a large number of organizations working together to get us to share internationally the data with all of these different projects. And the data that we've generated from research projects like I've been talking about is impressive, 10,000 genomes. But in the US alone, there are 1.2 million new cases of cancer every year. So it will be from routine clinical practice and genome sequencing, which is the future of cancer treatment, that most of the data will come, not from research projects. So we have to take that and use that data to study adult and childhood cancer. And while we're sequencing genomes, we have to study autoimmune diseases, congenital abnormalities, and infectious diseases by sequencing the infectious agent along with the genome. So all throughout medicine, we have the possibility of having truly an enormous amount of molecular information about the disease for the first time and engaging the scientific community to come up with the solutions that are needed. And they won't be simple. You cannot sit around and wait for somebody to come up with quote unquote the cure of cancer. It's very, very complicated. I've painted an encouraging picture at the end, but if you look into it in detail, every different type of cancer has a different way of evading the immune system, and we're going to have a very complicated solution to these things. When you have a huge and complicated problem with billions of data points, you have to share the data. When you need to get the data from the individual institutions, that's very hard, because currently almost every medical institution is just siloing that data. And you ask them, what about the data? And they say, our policy is no DNA sequenced by any of our patients ever leaves our hospital, period. That's the policy. Sorry, we can't share. That's not good enough. We have to get beyond that. We have to change, and the Global Alliance is trying to make this happen, from this picture of isolated data silos all over the world to a world in which there is an interconnected network of data sharing. So we're coming up with the application programming interface, data exchange schemas, and protocols that will make this possible, and our ethics and regulatory working group is in intensive discussion to make the social part of this work at this point. It's complicated. It's not just about getting everybody to put their genome in Dropbox, right? There are significant privacy issues here that have to be faced. There is a trade-off between personal privacy and contributing to medicine that you have to think about seriously, and we as a society have to contemplate that. The Global Alliance is now successful. We have more than 250 members from 40 organizations. That's 90 companies, so Google, Amazon, Microsoft, all of the biotech companies, pharmas are joining. Big institutes like the largest cancer center in the US, MD Anderson, and so forth, are joined. So we have the framework, we just have to execute on it at this point. And again, I'm hopeful. We can do this. As a collaborator, I had a hard time listing everybody we work with, and this is highly incomplete. But an enormous army of people have contributed to these projects to try to knit together the world's genome data and do something with it. Thank you.
Thank you, David, for the fascinating talk. Questions?
I should add that we are recruiting, so please come to Santa Cruz. Yes.
Hi, thanks for the lecture. I wanted to ask you, last night you mentioned, or maybe it was in the video they showed, the number 1 million cancer genomes, something
Yes.
that we might need 1 million cancer genomes. I was wondering if you could say a few words on why 1 million. What do you think could be achieved with 1 million cancer genomes specifically?
Yeah, well, okay, so, is it really 1 million and not 2 million and so forth?
No, sure, but that order of magnitude, what could be achieved with that order of magnitude?
Well, I think we get down, if you look at what happened with the Cancer Genome Atlas, which was roughly 10,000, you can actually plot something like the discovery plot that Michael showed about whether there's more stuff to learn if you keep taking more samples from the field, so to speak. And we're on the early part of it. You can just plot that. We have not saturated at all in terms of the significant mutations that we would discover. We're roughly we have a high confidence that we've probably found mutations that occur literally repeatedly, like 5% of the time in cancers. Unfortunately, the disease is so incredibly vast. If you do those estimates, there are thousands of other mutations out there that appear 1% or less that we don't know about yet. So that's the reason. The 1 million gets us roughly from 5% to 1% is another way of thinking about it.
Thank you.
Thank you very much, David, for a really magnificent talk. But a fair assumption is that most of the audience here are not cancer biologists. And they may not realize that, for instance, what kills cancer patients are metastases.
Yes.
And they may not know some of the history that, for instance, in 1997, Ruth Sager, who was a very famous cancer geneticist, wrote that she doesn't know many mutations, in fact she did it even stronger, that are associated with metastases. A similar statement was repeated by Fogelstein and Kinsler in 2004 in Nature
Yes.
Medicine, saying that again the progression of cancer to metastases is not associated with any mutations that he knows.
Yes.
So I think we should realize, and this is actually not the question, that at least some of the factors that are very detrimental for cancer patients, and some of them that may kill, are not DNA associated, but may be environmental, microenvironmental, phenotypic, whatever. And I think this is a very important lesson that especially the non-cancer biologists here should realize.
Absolutely. We haven't had a chance to look at the distinction between metastatic tumors and non-metastatic tumors at the molecular level with the large numbers that we saw here. All of the data that I was talking about, all the data generated by Cancer Genome Atlas and by the International Cancer Genome is primary tumor data. They started there just to get a foundation, not because they didn't believe that the metastatic problem was the key. It absolutely is. And the next phase of this has to look at metastatic tumors and do a careful statistical analysis of what you were suggesting, that there may not be as many mutations that are smoking guns for that transition to the metastatic state as there were smoking guns characterizing the primary state. There's a number of ways that might be true, and we might imagine that. Certainly, the transition in the primary state, you see a stem cell-like state for the cancers. They're growing and reproducing as if they were in a very early state of development. When they become metastatic and populate new tissues, they differentiate to adapt the molecular program of the home tissue. And so that is an epigenetic process rather than a mutational process. And so we do have to understand the epigenetic behavior of cell differentiation that creates metastatic lesions.
Maybe a question of mine, a question and a comment. this spirit of data sharing and tool sharing that exists in our field is sort of taken for granted, but I think it's really magnificent. And if you go to other disciplines, even nearby disciplines, this is definitely not the case there. And I think that part of the reason for the tremendous progress is this spirit and this culture of really sharing your data and sharing your tools, and only together we can really take a good stab at this data. But a big part is still not shared for some obvious reasons. For example, medical health records.
Yes.
For example, results of failed and successful clinical studies. Do you see a way to bring these two big elephants in the room into this game?
Well, we have a clinical working group along with a data working group in the Global Alliance for Genomics and Health, and they are trying to bring the pharmaceutical and university clinical trials system into a agreement on how to share the results of the trials and the underlying data. There are two basic problems: the privacy problems, but there also is the competitive advantage problem, obviously. And so we need to make sure that there is a reward for sharing in some sense that is clear to the people who have the data that need to be shared. And we also need to mobilize the patient advocates to demand that the research be enabled. And they are a very powerful force. The funding agents and the hospitals listen to the patient advocates quite strongly. So if the patients understand the value of data sharing and their medical institution is blocking it, then that will be a confrontation that might help loosen things up.
Okay. Yes, please.
Cyrus. The private companies or the private silos of data in the. So we have so on the data side we have medical institutions which are mostly nonprofit institutions, but still are not sharing their data for reasons of trying to protect the privacy of their patients or protect their competitive advantage or some combination of the two. In the software side, it's much better. So we have completely open, we're on GitHub. Anybody can go, you just Google Global Alliance for Genomics and Health. You can get into the GitHub dialogue. You can actually join a task team. You can start contributing changes to the code. It's a massive multi-100-person open source project, and all of the 90 companies have agreed that it will all be open source schemas and APIs. The companies are building proprietary software on top of this interface, which is one of the reasons they're deeply interested in helping with it. But it's the body of the interface is open source. And we have a REPRS implementation, which is open source as well, if you don't want to buy an implementation from the company.
Okay, let's thank the speaker again.